Hey guys, I want to talk about a new paper out this week from Baylor College of Medicine on a liver enzyme that helps to degrade proteins. Degradation is important. Proteins are constantly being made, and when they're done with their job, they need to be taken apart. They're no longer needed. Or if they weren't created correctly in the first place, they need to be taken apart as well. And this degradation is important. If they don't properly degrade, proteins can aggregate and cause all kinds of problems, including neurological problems. In this paper, the enzyme, the protein that we're talking about, degrades a protein when helps to degrade a protein when it doesn't fold correctly. So it wasn't created correctly in the first place. In that case, the point of the paper was that this liver protein has two functions. They've discovered a second function, which is kind of interesting. But I want to talk a little bit about this protein folding problem that it's helping to fix proteins that didn't fold correctly. What is protein folding? What's going on there? So this is a diagram of protein synthesis. This is how proteins are made in our cells. At the top you have the DNA double helix, and then there's a molecular machine sitting on the double helix that's going to make a copy. It unwinds the DNA double helix, slides along, and makes a copy. This process is called transcription. The transcript then is sent down to an editing step called splicing, and there another machine is going to splice it and edit it. That edited transcript then is sent to another machine, which is going to decode the information on the transcript and create this linear chain of ever-growing chain of amino acids. This process is called translation. Those amino acids, and there can be hundreds of them in a given protein, are then going to fold up. So there's your folding step. And there's machines there that help that process. And then finally there's a kind of a cleanup and recharging process that happens, so we can do it all again for another protein. This process here is very complicated. Each step here is very complicated. The idea that it would evolve by random mutations in the laws of nature is silly. It's not going to happen. This is a non-starter for evolution. Furthermore, in addition to the complexity, it's circular. You may have noticed this whole process makes protein synthesis. It also requires proteins. Each one of these molecular machines has a lot of proteins. There are many proteins involved in this process. So you need proteins to make proteins. It's kind of silly. From an evolutionary perspective, it's not going to happen. It's a chicken or the egg problem, right? I mean, you can read papers all about this, and they've got all kinds of speculations which really don't give you any details. But that's two major problems for evolution: the complexity and the circularity of this process. Another problem is proteins generally have multiple functions. Now, if you're you need to have a gene in the DNA that codes for this protein, that provides the information required to put together this string of amino acids, right? That's another big problem, because there's not just any gene is going to work. It's like the words on a page. You can't just type out any old words on a page and get an A on your essay. It's got to be particular sequences in the DNA that make a correct protein. And those are very rare. It's very difficult. The idea that evolution is going to create from random mutations these correct sequences is, again, it's a joke. But when you have multiple functions, it gets even worse. You might have two or three. We're now understanding that's the rule rather than the exception. Proteins have multiple functions. Very difficult to come up with the sequence for the code for the protein. So good paper, and the paper simply provides yet more challenges for the theory of evolution and why it really doesn't work. Until next time, religion drives science, and it matters.